For decades, the hardest part of plant biology was simply getting the data. Today, we are drowning in it. Raw genomic sequences are multiplying at a rate that outpaces our ability to physically process them. It wasn't long ago that if you wanted to do genome-wide analysis, you were restricted to a handful of foundational model organisms, like Eratabagus. That era is definitively over. This timeline charts the sequence wealth trajectory. In 2005, a few model genomes. By 2020, thousands of scaffolds. Look at the 2026 projection. We now access tens of thousands of highly accurate chromosome-level structures for previously ignored orphan varieties. In this era of structural sequence wealth, high-impact publishing relies on the precision of your analytical selection. Success is determined by the specific targets you choose to extract from the noise. Despite this wealth of data, thousands of labs globally are making the exact same error. They load standard automated software protocols to map classic, highly visible transcription factor groups, doing it over and over again. This chart illustrates the citation divide, the inverse correlation between a target's historical popularity and its actual field novelty. Notice the descending red line representing saturated transcription factors, like WRKY. Now contrast that with the ascending green line, tracking underexplored domains. Popularity does not equal impact. The immediate consequence of choosing those saturated targets is severe. Manuscripts lacking verified phenotypic novelty face an estimated 85% automated desk rejection rate across Tier 1 plant science journals, simply due to reviewer fatigue. Editorial desks are exhausted by basic coordinate listing. To survive, a submission must prove biological causality through multi-omics systems biology. Reviewers need to see how structure alters spatial function. To survive peer review today, researchers must abandon basic coordinate listing. Instead, you have to construct an objective, mathematical decision model to identify high-novelty targets and prove functional survival traits. The first step in building this model is pivoting away from classical regulatory families. Instead, you need to target the structural families that govern the actual physical cellular infrastructure of the plant. This table is the structural saturation matrix. It provides formal evaluation metrics for gene family selection based on target distribution dynamics. Up here in the red critical danger zone are the WRKY and MYB domains. The strategic action is clear. Avoid these completely unless you are pairing them with in vivo CRISPR pipelines. Compare that to the blue ocean frontier at the bottom. Domains like CLIP, Exocyst, and TOC-TIC remain virtually untouched, offering a maximum premium for clean discovery. Transporter architectures like MATE and SUITE dominate peer review for a specific reason. Abiotic stress tolerance is a crisis of cellular transport dynamics. These families map real physical infrastructure and dictate how a plant survives during severe climate events. We can visualize this strategic selection as a rigid hierarchy. At the bottom, you have a massive bottleneck of hypersaturated blocks, narrowing up to a very sharp peak of emerging frontiers. Targeting the complex internal logistics loops at this top tier yields a massive novelty premium. Almost no genome-wide tracking maps exist for these pathways outside a few select models. By strictly selecting explicit cellular transport and proteostasis machinery, you mathematically bypass the citation divide and guarantee a unique structural query for your manuscript. Targeting high-novelty gene families loses its impact if applied to an exhausted model crop. Heavy literature saturation in a species creates immediate competitive friction during review, regardless of the target gene. You need to shift your focus away from major cereals like rice and wheat. Instead, target understudied orphan varieties, like finger millet, that possess verified, real-world structural adaptations to local climate stress. However, you must pass what we call the infrastructure gate. You can never commit to a genome assembly based purely on the species name. This graphic breaks down the genome quality selection funnel. It is a quantitative filter to whittle down hundreds of public assemblies into a single viable project. To pass the validation rule, your target must survive the filtering sequences, specifically making it through the 200-plus chromosome scaffolds and the 80-plus anchored RNA-seq sets layers. Relying on contig-level assemblies is a critical error. They yield highly fragmented annotations, which generate catastrophic gaps in your subsequent domain alignments. This flowchart maps the algorithmic logic required to detect and reject those faulty assemblies. If you hit the config layer only, stop the execution loop immediately. Conversely, the green pathway confirms chromosome scaffolding is verified, leading to a safely cleared target scaffold. Enforcing these strict structural anchoring parameters up front prevents your team from wasting months of script generation on a dead-end genomic dataset. Even if you have the perfect crop and the perfect gene family, relying on basic linear 2D homology tracking alignments will no longer clear modern editorial desks. To secure clear publication space, you have to execute an architectural upgrade. This flowchart illustrates the multi-omics pipeline integration, moving your setup away from isolated profiling into layered functional biology. You start with the genomic space in step A, but then you must integrate deep SRA transcriptomics in step B and elevate the entire analysis to proteomics in step C. At that proteomic tier, simple script sequences are useless. You must utilize artificial intelligence to generate high-fidelity structural predictions. This graphic outlines exactly how advanced genome-wide analysis pipelines incorporate computational biophysics. The specific AI applications required here are mapping spatial configurations and interior pocket depths via AlphaFold 2, and modeling physical ligand binding affinities through molecular docking. By layering deep transcriptomics with AI-driven proteomic docking, your manuscript makes a critical transition. It moves from purely descriptive informatics to proving predictive biological causality. A common misstep here is designing massive, multi-cultivar pangenomic setups. These pipelines overload local workstation hardware, but frequently offer zero novel biological insight. Instead, project selection should be viewed symmetrically across multiple metrics, as detailed in the 7 axes of project viability. You need an ideal shape expanding across taxonomic novelty, assembly security, AlphaFold 2 feasibility, and climate stress relevance, rather than spiking on just one. This is the balanced optimization rule. A project with massive data volume but zero biological novelty will stall at peer review exactly as fast as a highly novel topic that lacks data depth. A truly publishable pipeline is one that optimizes your computational infrastructure limits and high-value functional biology simultaneously. Everything we've built addresses the final critical hurdle, hitting the fatal note from reviewers that the work is entirely descriptive and lacks biological validation. You do not need an active physical bench setup to provide that validation. You can secure robust in silico validation by relying on the deep multi-omics layers we just mapped out. This brings us to the Agrisys Project Clearance Checklist, the binary filter separating impactful science from automated rejection. The contrast is absolute. Abandon hypersaturated targets and contact levels immediately. Secure and prioritize high-novelty proteostasis targets, confirmed chromosomal scaffolding, and AlphaFold 2 docking loops. To clear the editorial desk, your manuscript must demonstrate how structural variations directly alter the plant's metabolic survival capability. Listing the variations is not enough. You must explain the survival mechanism. Replacing historical mimicry with an objective decision matrix ensures your research provides a unique structural map. By identifying these untouched frontiers, you establish a necessary citation for any future work conducted in that species.